Great pleasure to welcome our next speaker, Professor Marcus Buggert from the Karolinska Institute in Sweden. Marcus, thank you for joining us today. Marcus defended his PhD thesis back in 2014 at the Department of Laboratory Medicine at the Karolinska on the role of T cells in natural control of HIV infection. And then he moved on to working with Professor Michael Betz's lab at the University of Pennsylvania for his postdoctoral studies, where he pursued multiple projects, including the first identification and characterization of resident and recirculating memory T cells in the context of HIV and other human viral infections. Since 2018, he's back in Sweden, and he joined the Center for Infection Medicine at the Karolinska Institute as an assistant professor. His group focuses on studies of human circulating and resident memory T cells in the context of health and viral disease, including HIV, and of course, in the context of today's symposium, COVID-19. So the title of Marcus's talk is SARS-CoV-2-specific T cell responses in healthy individual and immunodeficient patients. So over to you, Professor Buggert. Thank you very much.
Right. Thank you very much. Can you all hear me?
We can.
Perfect. So I'm going to share my screen.
It's all good.
Can you all see my? Oh, good. Perfect. All right. So once again, thank you very much for the kind introduction. Thank you very much for inviting me to give this talk. So I'm going to talk, like you just said, I'm going to talk about SARS-specific T cell responses as everyone else in the session here, but focus more on health and also different immunodeficient states. And all focus of what I'm going to talk about today is going to be on T cell responses after mRNA vaccinations. It's only going to be on spike-specific responses, essentially. And the background essentially is that we know that mRNA vaccines, they induce robust T cell responses. Antonio and other people were referring to that in the past, that most of these responses, to at least to a very high degree, are taking place, of course, in tissues. And this has been close to my heart of what I've been studying. This is actually something that really these vaccines are doing extremely well when it comes to T cell responses, that I feel that they haven't got attention enough for, and that is that they're inducing an extremely robust, for example, follicular T helper cell response, which are necessary to generate good antibody responses. But nevertheless, what I'm going to do, like most of the other people today, is to focus on studies of the blood as correlates for protection from disease. So again, mRNA vaccines, they induce robust T cell responses, but the question that we have had now for a year is, how do these responses differ in healthy individuals versus immunodeficient patients? So there's different types of immunodeficiencies. One can say that for sure. So but broadly they can be categorized into two different kinds, one can say. Primary immunodeficiencies and secondary ones. So the primary ones are people which are born with for example, genetic mutations that affects maybe a protein that then in turn affects the immune system in different ways. And there's around 6 million people worldwide which are affected by primary immunodeficiencies. And then you have the secondary ones where individuals are born with a normal immune system, and these are instead caused by other factors, and it can be malnutrition, it can be viruses like HIV, which I've been working with a lot. Irradiation, chemotherapy, corticosteroids, leukemia, metabolic diseases. And this really affects hundreds of millions of people worldwide, as you probably can understand, given the fact that a lot of people worldwide have malnutrition or any of these other type of diseases which are affecting our immune responses. So why do we then care about studying immunodeficiency in general when it comes to COVID-19? And there's two different reasons for it. One is that immunocompromised patients, they have a higher likelihood in general to get severe COVID-19. Not all of them, but certain groups of them. This is from last summer. I still remember this when it came out. In the post-vaccination era, this number 44% came out as a share of the hospitalized breakthrough case patients in US who are immunocompromised. So basically, based on these numbers, so the number of hospitalized breakthrough infections were to high degree immunocompromised patients, and based on these numbers, FDA, CDC, and also here in Europe, in Sweden, we in August and September 2021, people who had moderate to severe immunocompromised states, they start to receive a third dose of COVID vaccination, and many people are now starting to receive even a fourth dose in certain countries. But maybe an even more pressing need to understand more about the immune responses in immunocompromised patients comes from viral persistence and the potential development of new variants of concerns. So we have known for a long time in people, when we have been studying HIV and other developing viruses, the viruses can mutate to a high degree, of course. But this is not maybe so common if you have a normal functioning working immune system for SARS-CoV-2, but it happens. And it particularly happens in individuals which are immunocompromised. And these are just some articles I'm showing now, which has been coming out lately, but it's an immense plethora of articles, which essentially has been coming out that all kinds of immunocompromised states are associated with long-term persistence and development and evolution of viruses and potential creations of these new variants of concerns. And I think to some degree, it's not so surprising, for me at least, it's not surprising that some of these variants of concern, such as Omicron, has been created in countries, for example, with a high burden, for example, of HIV and other immunocompromised states. And I think most of us, we really believe that the Omicron, it probably developed in an immunodeficient host or maybe a cluster of individuals. If that's due to HIV or other factors, we don't know at this point. But nevertheless, it's clearly so that unless we can stop the infection and evolution in these individuals, we will see more variants of concerns in the future. So what can we then do? Of course, we can vaccinate immunocompromised individuals. That's the most easy and straightforward way to protect them. But who of these individuals benefit from vaccination and who need booster doses? These are some of the questions that we are trying to understand more with the type of studies that we're conducting in our lab. So for about a year ago, what we did were to initiate this open label, non-randomized prospective clinical trial in which we assessed efficacy and safety of 2 doses of the mRNA vaccine from Pfizer and BioNTech in a huge group of immunocompromised patients and healthy controls at the Karolinska University Hospital. So these include patients which had primary immunodeficiencies, all kinds of them, I can say. I'm not going to have time to go into all of them, but it ranges from CVID to more of these monogenic diseases. Individuals which has HIV infection, hematopoietic stem cell transplanted patients, solid organ transplanted patients, and also people with specific hematological malignancies, such as chronic lymphocytic leukemia. This is an ongoing collection. I'm mostly going to show data now between day 0 to 6 months after vaccination. But again, it's an ongoing collection where we're right now studying responses after 3rd dose and which impact the Omicron wave has on, for example, T cell and antibody responses as well. I will show some of that data towards the end for some specific immunocompromised groups also, for example, how a 3rd dose can have an impact on SARS-CoV-2-specific T cell responses in some of these individuals. But this is data, then antibody data from a publication that was published by from our consortia earlier this year, or sorry, last year, 2021 in eBiomedicine. It basically shows that antibody responses to vaccination differs considerably between different patient groups. And what we have essentially done here is to look at the antibody response between day 0, 10, 21, and 35 in all these different groups, and to look at the seroconversion at that time point when we really think that most people would have seroconverted. And in particular, what we can see is that solid organ transplanted patients, they are the group that show really the lowest vaccine responsiveness after 2 doses of mRNA vaccination. This is not the first study which has shown this. This is just a summary which I'm showing now from a news article, which kind of summarized all the different studies which essentially out there and put a median for the number of patients which has a seropositivity rates. And you can see from this, just like our study, that solid organ transplants, they are in the bottom, so they respond in general worse to vaccination, and followed by hematological malignancies. And we actually see the same thing, that people with different type of chronic leukemias, they respond poorly to the infection. But some individuals, such as HIV infection, particularly those people which are treated, I should say, with HIV, they respond pretty well, for example, to the vaccine. But the question again is that what most people are looking into, of course, are antibody responses, but what we were interested about was to look also into how the T-cell responses were developing in these individuals to see if they showed other type of patterns in some ways in comparison to seropositivity. So this is essentially summary of the slide of the data after 6 months, and it's a fairly busy slide, but I'll just go through it and we can focus to some degree here on the flow cytometry pictures. So this is just a normal AIM assay for CD4 positive T cells. We do a overlapping spike stimulation of the full spike against the wild type, and we measure through AIM markers CD69 together with CD4 ligand, which is CD154 here. And you can see after this is at day 35 at the peak essentially of the response that healthy individuals, they induce a fairly robust response. But many of the other individuals in the other immunocompromised group, just like the antibody response we're showing, are actually inducing pretty good responses as well. But the biggest difference here probably is that while the healthy controls, all of them are responding well, you can kind of see that in all the different immunocompromised individual groups, you can start to see that there are always some individuals that do not really respond well, also when it comes to the T-cell response. And it differs a little bit depending on which group and what kind of treatments they're on, which I don't have time to go into any details of. But what I want to emphasize on again is that just like the antibody responses, we do also see that these individuals which are solid organ transplant individuals, as well as chronic leukemia patients, they are the ones that, in addition to having a poor antibody response, also have a poor T-cell response. And this is kind of summarized here as the peak of the response is day 35, where we're looking again into the magnitude of the CD4 response, where you can see that SOTs, which is this group here, they have, in comparison to healthy donors here, are the individuals which has the lowest magnitude of responses, but also the chronic lymphocytic leukemia patients do have a significant lower response. And we can see with time, some of these individuals here, when we're starting to approach 6 months here, actually have either potentially got infected or got a third dose, but potentially some of them got infected. But in general, what we can see is that the SOTs, they are lagging behind the other groups also when it comes to the T-cell response. It's not only antibodies in general. And the reason for that is quite simple, because these individuals have put on very heavy treatments that block cell proliferation in general. So it blocks both B cells and T cell proliferation, and that's the reason why they don't reject those organs, but that's also the reason why they don't respond well to the vaccines. So what we thought that we could do as well, we're still to say that, okay, so it looks like there's a correlation, and it is a correlation between the antibody responses and the T cells. But what we decided to do were to look at the peak of the response, then to say that, okay, but are there some individuals still that potentially do have T-cell responses that are seronegative? And we could indeed find that there was some of these individuals that were in this quadrant, so to say here, which were T-cell positive, but they didn't have any antibodies. These are actually individuals in these bar graphs here, which are in yellow. So you can see that there are some SOTs, there are some CLL patients. In general, they have lower magnitudes of the responses if they're positive, but nevertheless, there are some that are reaching actually a threshold. But there are some interesting patients also in this group here, which has T-cell responses but lacking B-cell responses that we were interested to study a little bit further. And some of these patients are within this PID group, and they're known as patients with XLA, X-linked agammaglobulinemia. And these are patients which are lacking a protein known as BTK, which is important for B cells to mature. So these patients, they have no B cells, they have no mature B cells. So essentially, they function as a human knockout model to study the impact of what B cells will have on, for example, T cells or maybe other factors as well, to understand more, to gain mechanistic insight into all kind of questions one can imagine. So we thought it was interesting to follow these patients versus healthy controls just in more detail to understand what will the impact of a T cell response be if you're totally lacking B cells. So in order to understand this a little bit more in detail, what we decided to do were to do 10x genomic single cell sequencing analysis. So we took blood from 3 healthy donors, 3 XLA patients. Both of them generated good T-cell responses. And this is something, again, I can say that they do induce robust responses, even without B cells. We looked at 14 days and 6 months post second dose, did peptide stims. We oligoconjugated these cells with antibodies stainings. And then we looked, and then we sorted cells based on AIM assays to look at the functional profile through single cell RNA and TCR sequencing. And I'm not going to have time to show the details of all these analyses because that would probably take the entire session to show all the data that can be generated. But this is just to pinpoint 2 points that one can say that we can answer with these type of questions here by just focusing now on the CD4 data. The first thing is that through these type of technologies, you can kind of see here for the UMAP is that you can create different clusters. But in contrast to what people, including ourselves, when we're using conventional flow cytometry and we're identifying classical Th1 or Th2 or TFH archetypes, we don't really see that you have IL-2 or TNF or interferon gamma production being in one specific cluster. It's more like a continuum where some of them are merging together, and actually IL-21 together with gamma and TNF, for example, can form specific clusters here around this blue belt here in cluster 1. But on the other hand, we do see that some other clusters of IL-22 producing cells, for example, or IL-17 producing cells, etc., they are more associated with other clusters. So it's a continuum of different cell clusters that doesn't necessarily overlay with what we know as TFH, Th1 cluster when you start to do an unbiased type of clustering analysis like this. The second thing I want to highlight, again, comes back to the point about the XLA patients here. What type of response do they generate? Is it poor even if they have a T-cell response? It's actually not. If anything, what we do find is that their responses are as functional, maybe even more functional in general. We can show that by a volcano plot here and actually see that some of the genes, normally even which are associated with TFH cells, are actually highly induced by these individuals. So altogether, we can say that we can use these type of patients to and unique technologies to understand exactly what type of response that they're able to induce. And we can see that these patients totally lacking B cells are doing fine T-cell responses, which of course could be good to know in other type of vaccine settings if you are lacking B cells, for example, if you're treated with rituximab or CAR T-cell therapies in cancer therapies maybe. Okay, so that's that part, but I thought also I'm going to just touch upon this point about Omicron recognition that we talked about in the beginning. So we now know the SARS-CoV-2 specific T cells, they cross-recognize variants of concerns like Omicron pretty well. This is a study that we published early this year, focusing in here on CD4 responses, comparing the wild type response against the Omicron response. And essentially through the same assay, we can see that there is not a big difference if you look at the total full spike specific response between wild type versus Omicron, not between vaccinated, not between convalescent individuals. We can do the same for CD8 positive T cells. So now we're coming in on that arm a little bit here now as well, but we're using a little bit different markers like CD69 together with CD137, which is 4-1BB. And we can see that there is some reduction here against Omicron. In vaccinated individuals is 8%. It's a 30% reduction in convalescent individuals, but in general is nothing comparable to the reduction of what one would see with neutralizing antibodies, at least when we're looking at the full, so to say, spike specific response. So with that said, though, what we have decided to do now is to continue with some of these analyses and look also into immunocompromised patient groups. So for the sake of time, I'm just going to focus on one of these patient groups now, but it's in general, the same applies to the other immunocompromised patient groups as well. So when you start to look into, for example, PID, primary immunodeficient patients, you do see that same type of principle as we just shown recently in healthy individuals. When you start to look into the full spike, wild type versus Omicron, there's very little difference between the responses. It's just slightly lower, and there's no significant difference really. But when you start to, however, look into those Omicron mutated regions, and you now create epitopes which are homologous for the wild type, you start to actually see that there are some differences there. So it could still be so actually that Omicron have an impact, but it's just that it's masked by the responses by all the other epitopes by T cells when you're looking into the full spike-specific response. So you do actually see, if you, for example, look at the stimulation index for CD4 plus T cells, that there is a reduction of Omicron mutated spike-specific epitopes if you compare it to the homologous spike response by the wild type. For the CD8 responses, they're a little bit lower in general, the responses, so we do not see too much of a difference here. The number of positive responses here are lower in general, and I think that's the reason why it's not significant. But there are some trends that kind of points towards this direction as well, even if they're not as clear as for the CD4 responses. But nevertheless, this is after 2 doses of vaccines after 6 months. So the final question that we had was to look into what happens when you give people which has already even had an infection, and you now give them a 3rd dose in patients with chronic leukemias. So when you do this and you look into the CD4 response versus wild type and Omicron, same assay as before, it's just a normal AIM assay, you do see that there's a slight increase after a 3rd dose for the CD4 response, but it's not significant versus what you see post-COVID and also before the 3rd dose. Again, same here, the Omicron response is pretty good. This is against the total spike response, so this is not against these specific mutated regions. But when you start to look into the CD8 response, there is actually a quite dramatic increase in the number of CD8 responses, both against wild type and Omicron in these patients after a 3rd dose. And this is something that has been seen in multiple sclerosis patients as well. And it really emphasizes this point that with joint antibodies, it could be so that people will maybe benefit from getting a 3rd dose by boosting their CD8 responses as well. So the summary of my talk is that specific immunocompromised patients, such as solid organ transplants, they display not only reduced humoral responses, they also have reduced cell-mediated immunity, which is accompanied by reduced functional spike-specific T cell responses, which I didn't go into too much in detail. But it's not only so that they have lower responses, they have less polyfunctional cells in general. Patients with an inherited lack of mature B cells, however, such as these XLA patients, they exhibit detectable and functional spike-specific T cell responses despite no mature B cells. And we can show by single-cell RNA sequencing that the mRNA vaccination, it really induces this broad set of spike-specific T cell phenotypes that goes beyond classical T helper and memory characteristics in both XLA and healthy donors. We can also see that T cells, they cross-recognize the Omicron variant also in immunocompromised patients, and that a 3rd dose really boosts the CD8 response to a higher degree than the CD4 response to these higher levels in immunocompromised patients, such as in chronic leukemia. So with all this, I would like to thank all the group members here in BOLD, and also other collaborators at the Karolinska Institutet and on the hospital side, as well as patients, healthy volunteers, and our funders. So thank you very much, and I'm happy to take any questions.
Thank you very much indeed, Marcus. That was really very interesting, and I think a fantastic insight into just helping us to understand a little bit about the best way to help and to understand the immune response of the immunocompromised individuals, because I think it's very clear that the immune response is very dampened down. And I guess one question, and you sort of touched on it on the end there in regards to the observed increased CD8 T cell response, was that what's the best way of going to be able to be treating these patients? Is it going to be really just focusing on trying to get as much CD8 response as possible, because that's kind of all that we see happening in extra boosters, or what are your thoughts there, particularly in the solid organ transplant?
Yeah, it's a great question. Maybe it could be a combination of that, because many of these CLL patients, they are on these type of treatments which are blocking mature B cells from developing, because it's a B cell disease, it's a B cell malignancy. So that reason they are on these new generation of BTK inhibitors, which are blocking mature B cells from forming. And I personally think that they will still gain probably therefore from getting a 3rd shot, but also what clinicians are giving these patients are monoclonals as well, so that they have protection through them as well. One of the issues, of course, that have emerged now with Omicron is that many of them are not working as good anymore, but there are some of them which are still working pretty well. But this is good points. These are ongoing discussions. We had discussions with the clinicians about this today, essentially, if it would be beneficial to put these individuals now on third and fourth maybe doses as well, because we are boosting the CD8 responses. So it's an ongoing discussion, I guess, that is going on like really right now between us researchers and the clinicians as well at this point.
Yeah, no, very, very pertinent. So thank you. That's great. Any other questions at all?
Yes.
Antonio, you got your hand up.
Hi, Marcus. Very nice presentation.
Thank you.
Very short question. I mean, clinically speaking, the subjects that are B-cell deficiency and they are only T cells, do we know something what's happening if they're getting infected?
Yeah, right. So some of them do get persistent infections, but I can say like this, that these patients, for example, that are totally lacking mature B cells, they usually don't have problems with respiratory tract infections, including SARS-CoV-2. They usually have problems with bacterial infections to a high degree and some enteric infections as well, but not really with SARS-CoV-2. They usually don't develop severe disease. One of these patients, for example, was the fourth patient actually here that we couldn't include for the RNA sequencing, got infected because the person didn't develop any antibodies, but literally had good T cell responses, but didn't get any type of severity of the disease. So it's interesting. It's an interesting group of patients, of course, to use to study many of these different aspects, as you are, of course, aware of as well. But it's a good question, and you're of course aware of it, that other people like John Barry and others have shown that more robust CD8 responses and the lack of B cells is associated with better prognosis, usually in malignancies and stuff like that as well. So I kind of think that it kind of points again towards that the role of a cell-mediated immune response being important in these patients.
Great. Well, thank you very much indeed. Thank you, Marcus, for your presentation, and do join us for the panel discussion after the next speaker, if that's okay. Fantastic.